So thank you, Sheena, for this great introduction. I couldn't have a better introduction for the amyloids. And first of all, I must admit that I always loved in protein structure-function relationship and dynamics since my days doing a PhD with Nir Ben-Tal. And to be honest, I thought that fibrils are pretty boring because they are either scaffold proteins or they're just kind of end products of different things, and I found out that they are so not. They are very, very interesting. They are very different. They are fun. Just one example, Sheena also showed all this polymorphism, meaning that one structure can give you very different, one sequence can give you very different structures of fibrils. So you already understand that it contrasts the dogma of Anfinsen of one sequence, one structure. So fibrils are challenging and exciting in different ways. And so I'm not going to focus on disease-related fibrils, but I'm going to talk about three different types of fibrils. Ones that are involved in the biofilm structure, and I will explain what it means, and fibrils that are actually killing cells, either bacterial cells or human cells. And this field was tremendously boosted of these functional fibrils, where in 2002, when Matt Chapman discovered that fibrils that compose the Enterobacteria biofilm share biophysical characteristics with the human amyloids that are involved in devastating diseases, and you heard a lot about it from Sheena. But I want to remind you something that is another special feature of these fibrils. They are highly stable. And what does it mean? I think maybe the most pronounced examples are the prion diseases. For example, we know them mostly as the mad cow diseases that happened in the 90s, or Yakov-Creutzfeldt in humans. And the first human disease that was a prion disease was named Kuru. It was found in a tribe in Papua New Guinea of people that are eating human flesh, including the brain. And they contracted this neurodegenerative diseases by eating the brains of others, and then the cows had the mad cow disease, and then the Yakov-Creutzfeldt disease. And what is so special about these diseases, they are infectious, but there is no microorganism involved. There is not a virus or a bacteria or a parasite, nothing. And Stanley Prushner reached this amazing conclusion. I think that he got a lot of resistance at first, that we are talking about transmissible proteins. And how is it even possible that you can contract disease by eating a bad protein? So think about this. You are eating meat. So it means that contaminated meat. And now we know, like, foie gras, for example, contain these things. So don't eat foie gras for different reasons. But you're eating contaminated meat, and the proteins sustain the entire way through your stomach and the acidic environment, and all the proteases and bacteria and whatever. And somehow they find their way to the brain, and they're causing diseases. So it means that the structure of this protein must be special, right? So I think that one of the main motivation to find the structures of these prions that are similar to amyloids, or they are amyloids, depending who you ask, is to understand what can give you this amazing stability that you can chew a protein, you can eat it, and they still get to the brain. So back then, there was not a lot of options and to get structures of amyloids. Because most of the fibril can be, so it depends on which one, but some of these are very, very long fibrils, and they have structured backbone, but a lot of disordered regions that are completely disordered, or they have some native conformation, but it was impossible to determine structures. So the idea was developed, and Udi Gazit sitting here was one of the people showed it first, that few residues, it could be 4, 5, 6, Udi showed it even less, even one amino acid, can hold the spine of this amyloid. And it's also recapitulating several of the biophysical characteristics of the amyloid. So maybe we should work on only the spine. So a lot of, Sheena showed some methods that were developed to recognize these amyloidogenic spines. And David Eisenberg managed to form crystals of these short segments. And the nice thing that they form crystals that we actually can solve their structure, but it's very, very difficult because these crystals look like fibrils and are very thin and are very flexible and they have a lot of issues. And so you can use very limited number of synchrotrons for this. You need microfocus beamlines, and the ones that I tried that actually worked were Aragon in the US and ESRF and EMBLP14 at Petra 3 at DAISY. And it's a very dedicated methodology to solve the structure. And David Eisenberg lab tried for 10 years before they succeeded at first, but they did in 2005. And the motif in all of this fiber-like structure is a pair of beta sheets formed from segment stacked in registers. So what Sheena was talking about, the cross-beta steric zipper structure, this is how it looks like. So you see the stacked layers and amino acid protruding from the beta strand from this tight, dry, complementing interface, which David Eisenberg called the steric zipper. And you can imagine why, because it looks like a zipper, and then you have hundreds of thousands of layers like this across the mating beta strand. So this is a very stable structure, and it manifests the stability of the amyloid fibrils and their self-seeding characteristics, so you can understand the prion diseases in a sense, because if you get something small like this in your stomach, it doesn't dissolve. It's you can put it in crazy solvent in the lab, and calling them lab killers, not the students, but columns. And then it meets a protein that is similar to it, so you have another layer, another layer, another layer. So it's sequestering the polymer like this. So it explains a lot about these diseases, and the structure explained all of the information that was taken from different amyloids, even in the 70s and 80s, and gave them a structure of the cross-beta. And as Sheena mentioned, now we have more and more structures of fibrils coming from also mainly now cryo-EM that give us much more information. But I didn't at all wanted to talk about the human amyloids. I wanted to talk about amyloids coming mostly from bacteria, but not only. So the first thing that I want to show you is our amyloids that form the scaffold of biofilms. And biofilms are agglomeration of bacteria. They secrete a lot of things outside. It can be proteins, DNA, polysaccharides, whatnot. But at the end of the day, they form something very, very sticky and very, very stable that can bind to human tissues or to pipe. And think what happens in the pipes beneath hospitals when all the resistant bacteria, as Ada described nicely, but all the antibiotics of the hospitals are going together to the same place. So what kind of bugs are growing there? And Adam mentioned the resistance problem, but biofilms don't even let the antibiotics penetrate. Okay, so you cannot treat a biofilm-related infection. You just cannot get rid of it. If you're unfortunate to get a biofilm-related infection, it has to be surgically removed, and if it's implants, they have to be taken out. You cannot treat biofilm-related infections. And when we started the work on microbial amyloids, there was no structural information available, and we didn't even know, are they similar to human amyloids, but they're from bacteria, so what are we going to see? And at the beginning, we used the Eisenberg approach because the amyloids involved in biofilms are hundreds of amino acids, and they are not ordered at all, and you can determine the structure with crystallography. So we used the Eisenberg approach to go for this most amyloidogenic segments. And every time that we solve the structure of an amyloidogenic segments from a biofilm-related protein, it can be from different bacteria, we got the steric zipper structure. So as a crystallographer, I was happy that the first time that I saw the structure in my own lab, but at the end of the day, it was a structure that I saw 200 times before in the Eisenberg lab. But it was the first time that we see that also bacterial sequences can form these steric zipper features. So we can show that this tendency to form these kind of structures go from bacteria to human, and this was exciting. And then we thought, what are the implications of this structural similarity? So if the biofilm-related amyloids are similar to the human amyloids, what does it tell us? So we know from our protein world that if you have a similar structure, maybe they will be inhibited by similar inhibitors, right, like kinases. So why not to try? And we collaborated with Dieter Wilbord in Jülich and Düsseldorf, and they had molecules already in clinical trials for Alzheimer's. And okay, so we have a similar structure to amyloid beta in Alzheimer's. Let's try these molecules. And we tried several, and we found 2 compounds that inhibit the fibrillation of the biofilm-related amyloid in Enterobacteria, like E. coli and Salmonella. And we did the inhibition assays using different kind of methods, but I just want to show you one. So these proteins are so crazily stable that if you put them in SDS buffer and you boil them to 95 degrees, not just only that the fibrils are not dissolving, the fibrils they keep on growing. So this is why I call them like leg killers, because every column that we use, we had to throw away. I don't know how much money we spent with Daniel Biotech replacing columns. So now my students know how to deal with this. And people also ask me about safety levels. So these proteins can be dangerous, of course, so I advise my students not to eat them or not to inject them. But I think that otherwise we are pretty okay. But when they are in the powder form, you don't want to breathe them, definitely, because they can go into your lungs. So not breathing, not eating, and not injecting, you should be fine. They're not going to jump at you otherwise. So think about this. So if they are not dissolved in SDS, we can actually use it to test inhibitors. Because if we just let this protein fibrillate, so it doesn't run on the SDS page, and we don't see it. But when we add the inhibitor, you can see it running for a longer time. It's soluble for a longer time. So this is one of the properties. But I think maybe the coolest thing was that these 2 compounds also reduce the biofilm biomass of Salmonella. And think what it means. We repurpose drugs from Alzheimer's to use as anti-biofilm based on structural similarity that we discovered for amyloidogenic regions. And I think this is pretty exciting, just as a concept of repurposing drugs like that. And these are compounds that are already in clinical trials, so they are being already tested for all the pharmacokinetic properties and so on and so forth. So this was the one implication that we have from the structural insight. And the second one, okay, so if they have a similar structure, can one affect the other, like in prion diseases? Because we are, in people that are eating human flesh, they are being, so it's a human protein that is templating another human protein, but in the mad cow disease, in the Yakov-Creutzfeldt from the contaminated meat, it's coming from a cow protein and affecting a human. So there is a species barrier. So what happens here? And we showed it only in vitro that seeds from the bacterial amyloids can facilitate the fibrillation of the human amyloid. And this is, of course, only in vitro result. And we showed nothing, we tried nothing on a human level or mice models. But it makes you think, what's the role of microbes in neurodegenerative diseases? And there is a lot of buzz recently about the gut-brain axis and similar to prion diseases, what happens with our microbiomes, how our microbiomes affect the propensity to get neurodegenerative diseases, and what happens with very aggressive infections. So I'm not telling you that every bug that you encounter it will give you Alzheimer's. I let my kids eat from, whatever, but I think it's important to be exposed to bacteria. But we have to think about this possibility when we think about the propensity for neurodegenerative diseases, because no one knows why some people get Alzheimer's and some people don't if they don't have genetic mutations. So this is what to sense about this relationship. And now I want to move to my kind of favorite bacteria, because I had a long and intimate relationship with this bacteria 20 years ago. It's Staphylococcus aureus, and it secretes a family of peptides called phenol-soluble modulins, PSMs in short. They're a family that are highly virulent peptides. They participate in many of the pathogenicity of this bug. And some of the very virulent Staphylococcus strains, 60% of the protein mass they secrete are PSM, so they are definitely important for this bacteria. And we started working on them because Blaise Boules' lab suggested a few years ago that they might form amyloid. So we started with two members of this family. You can see they're short peptides, about 20 amino acids, PSM alpha 1 and alpha 4. And Blaise Boules' lab showed that they are building the scaffold of the Staph-Aureus biofilm. And near in the lab, we're sitting here, showed that they form these beautiful fibrils, and they have a cross-beta diffraction and the structure of the most amylogenic segment, because we couldn't crystallize the entire length. Indeed, it's a steric zipper. So this was another example of biofilm-related amyloids that formed the cross-beta steric zipper structure. But why am I telling you this? I'm telling you this because we found a huge contrast in this family of peptide, because this one, alpha-3, really surprised us. They are homologous sequences, and they are being secreted together from the same operon. And this one, alpha-3, it's known to be, it was never found in biofilm, but it was known to be the most cytotoxic member of the family. And we didn't know if it forms amyloids or not. So Einav in the lab, Einav was not doing a postdoc, she showed that it forms these beautiful fibrils, and it binds the amyloid indicator dye, tau-flavin T. So far so good. We thought it's another amyloid, okay? Maybe interesting, maybe boring, like whatever you think about it. But that's something where the biophysics didn't add up. Because in amyloids, they can start from disorder, they can start helical, they can start beta, they can start whatever. And when they form fibrils, they become cross-beta, so they become beta-rich. But in this case, looking at the secondary structure of the fibrils using solid-state CD or FTIR, Einav observed that they are still helical also in the fibril state. So what is going on with this peptide? And somehow we tried, I don't know why we tried, but we did, to crystallize it. So I'm saying why, because in David Eisenberg lab, we never crystallize something that is more than 8 amino acids, we're usually 7. And this was the full length, the entire 22 residues. So I was shocked that we got crystals that look like amyloid. I thought that my student did something weird. And I sent the crystals to mass spec several times just to see that we have the full length, and it was. So we fought for this structure for a very long time, and many synchrotron trips were involved and many hours of processing, and it was worth it because we solved this structure. So at first sight, it looks like the canonical cross-beta structure. Two mated sheets, individual molecules are situated perpendicular to the fibril axis, just as the cross-beta, but the individual molecules were helical, not beta strands, and this is the reason that we couldn't crystallize it, because it was structured all along the sequence. So obviously we are very excited, and we named this one cross alpha from obvious reasons. And so if we compare cross alpha to cross beta, you can see that the quaternary arrangement is very similar, but is composed of a different secondary structure. So it's a new level of polymorphism. It's a new level of polymorphism at the secondary structure level, not in the tertiary structure level. And they have a similar quaternary structure. So I think it's amazing. And the question is to call it an amyloid or not. So you leave it like for each one of the two decide. And but for me, it forms fibrils. They are unbranched. They bind the amyloid indicator dye tau-flavin T, and they have the individual molecules situated perpendicular to the fibril axis and mated sheets, just like as the cross beta, and no other fibrils is like that. And I think even the concept of seeing these helical sheets is pretty amazing. So obviously we were super happy about this very cool structure, but you know, you want to publish it, so you also need to do what you need to say something about what this fibril is doing. Is it even relevant? Is it biologically relevant? Like, does it have a function? So we knew that it's the most cytotoxic member of the family. I'll skip this one. And this peptide is a natural killer. So these are T cells, and after 5 minutes of incubation with this PSM alpha 3, you just see a graveyard of T cells. So this is a killer. Now the question is the monomers killing what is really killing the cells? So think about in biochemistry, when you want to, you solve a structure and you want to know if the crystal structure is even relevant, you use biochemistry, right? You do mutations and you see how the mutations in relation to their location in the structure affect the function. But here, it's very complicated because what about fibrillation? If it kills fibrillation, so it kills the entire structure, and so it's a structure, it's a fibrillation, so you have another layer of complexity. So what we did, I won't go you through all the details, like it's not necessary, but we did a series of many mutations, and she tested the ability to form fibrils, the secondary structure of the fibrils, the biophysical properties of the fibrils, the stability, so on and so forth, and the toxicity level. And from all of that, we came to the conclusion that fibrillation is needed for toxicity. We cannot say that it's the fibril, but we know that if you lose the ability to fibrillate, you are not toxic anymore. So this was our conclusion. But then Einav went on looking at the mechanism a little bit more, and you can see the aggregation of this peptide on the cells, and she also colored the membrane, so it's very obvious that there is co-localization with the membrane. And now our model of toxicity is that people in the amyloid field are always looking for a toxic entity, oligomers or fibrils, and I don't know about the rest of the amyloids, but what I think for our cross-alpha amyloid is that what happens is that the membrane is not a naive part in this game. So once the peptide sees the membrane is inducing helicity and aggregation, and this we know because we measured with Razi-Elinek and VGU. And then there is a lot of aggregation, and we think that the aggregation is involving the lipids, so there is co-aggregation with the membrane together, and this is how it chooses the membrane is killing the cell. So we are not talking about toxic entity, because if you don't have monomers, you won't have toxicity, because you need the monomers for this dynamic process. But if you cannot form the fibrils, you also don't have toxicity. So you need the ability to form these cross-alpha fibrils, not fibrils or monomers, the ability to form these cross-alpha fibrils. So this is our model for toxicity, and if you ask me, I think it also holds for some other canonical amyloids, but I have no results about that, so I cannot really say. But think about this crazy family of PSMs. The bacteria is secreted these family members, and 2 of them are structuring its biofilm. One of them is killing our immune system, so we cannot fight the bacteria. And moreover, these peptides are being chewed in vivo into active derivatives. And Nir here, sitting here, found that this one that has antibacterial activity also have this atypical kind of amyloid, non-amyloid structures. This is a hexameric beta-rich structure. So this bacteria is crazy because it forms different structures of fibrils for each different activity that it needs. Okay, so it's a very diverse and interesting kind of phenomena. And we were asking ourselves if the cross-alpha is a one-time thing, and obviously it's not, and we were looking for more of this thing. It forms from alpha-hepatic helices. So we found that an antibacterial peptide secreted by this Australian toadlet, so it's like little toad. I will call it a frog if I may. So this peptide also forms cross-alpha structure, and Nir showed how it caused deformation of the bacterial cells. And he also showed that if you try to fibrillate this peptide in very low concentrations, you get fibrils, but they're very scarce. So you basically don't see many fibrils. But when you use the same incubation time in the same concentration with the bacterial cells, then you have massive fibril formation that are covering the bacteria. So I think it's again the same game. The bacteria is affecting the fibrillation, and then the fibrils are killing the bacteria using this toxic mechanism. But then Nir found something really cool. I think it was very exciting, I think even more than the structure in some ways, because he found that this peptide is a chameleon. So forget biophysics, okay, don't look. But I will summarize. So what he found is that if you incubate this peptide with no lipids around, just in buffer, it forms okay, let's start. If we incubate this peptide with lipids or something like this, then the peptide is helical, okay, and it forms CoROS alpha, and it did FTIR and solid state TD and solution and fibril diffraction. So with the lipids, is helical. If you incubate it without the lipids, it's cross beta, okay. So what I don't have your nice cartoon. But Nir made this nice cartoon that I'm the frog, and I'm secreting the peptide, and it's probably monomeric. And if I don't see bacterial cell around me, maybe I'm becoming a cross beta, and he showed that cross beta are likely to be not toxic, and I'm just sitting there. But if some bacteria comes and it sees the membrane, then it induces helicity, induces cross alpha aggregation, induces toxicity. So it's really fun, and Nir showed it with a lot of ways to show this back and forth transition, and also thermostability, because the cross beta, I told you, they are super stable, so if you hit them to 60 degrees, you still have fibers. The cross alpha are not that stable. Cross alpha dissolve. But this guy, the frog guy, stays stable, and we think that when it stays stable in high temperature, because the heat drives it to cross beta, so then it's stable. And when the frog is sitting in the sun, then maybe it's cross beta. When it goes into the water and everything is dissolved and you have a lot of bacteria around, maybe then it's cross alpha. But we really don't know. We just can make models and assume and have fun with frog schemes. And I want to finish with the last one. Since we were doing antibacterial peptide, we said, why not human? Let's use humans antibacterial peptides. And ITSIC solved this amazing structure. And I'm not claiming it to be an amyloid by any means. People don't need to be concerned about that. So it's a fibril composed of bundles of helices that are densely compact. It forms this hexaberry configuration with a central core. And if you look on these colored bundles, so there are 4 helical bundles with a hydrophobic core, and they are very tightly packed. And interactions between the bundles are stabilized by a network of polar interactions. So you have a network of polar interactions and hydrophobic packing, and it's a very stable structure. And if you just color the surface by hydrophobicity, so you see this zigzag belt of hydrophobic, and they're actually not polar, they're actually positively charged belt. So you can imagine how this thing interacts with negatively charged bacterial cells. So, and this fibril is also very stable in solution. It forms these beautiful wide ribbon fibrils that are pretty stable, but when they start dissolving, you can see that they are composed from tiny fibrils that laterally associate, and you can see it also in cryo-EM images. But I think for us, the most important thing is the interactions with the bacteria. So here you can see these beautiful ribbons, how they interact with the bacterial cells, killing it. And I think it's super. So we know that the fibrils are interacting with the bacteria, but do we know that the crystal structure is relevant? We don't, right? So we do a series of mutations. And just to put it in brief, when you mutate residues that are highly buried in this assembly, you lose self-assembly, and you also lose antibacterial activity. If you mutate the most exposed residue in this bundle, the residue with least contact, even if you change from polar to hydrophobic, you still have this nanostructure interacting with the bacteria. So, you know, at least from the mutations that we try, it seems that the structure is relevant. Now, what's the next step for this kind of thing? Do I want to solve more and more and more structures? Yes. Is it the right direction to go? Maybe. But what I really want is that I want to see this interaction at atomic resolution. And I want to see the toxicity process at atomic resolution. So I cannot crystallize something with bacteria, but I can't use cryo-EM for this. And I started a few months ago a sabbatical in Hamburg in DAISY, where also Ada went, so I'm kind of following good steps here. Big shoes, but it's fun. And I'm affiliated with the EMBL, and I'm working with Kai Grunwald, and these two amazing people. They are the head of the facility of the cryo-EM and light microscopy. And the idea is to solve structures of these fibrils not in crystals, and to do correlative light and electron microscopy. It means that you can, with light microscopy, to see the interactions of the cells with the peptide, mark the location, go with the same grid to the electron microscope, and look at high resolution what is happening there. And this is pretty cool. So I'm really enjoying it, and I think I can do really nice things there at the CSSB. But what happens when I go back to Israel? Can somebody tell me what to do? Because we have nice microscope in Weizmann, but there are only 2 for the entire country. We have nice microscope for material sciences and chemical engineer at the Technion. We have nice old microscope that some people here made amazing results with Grutaa. And we need a national center for electron microscopy that we can do the single particle reconstruction, we can do correlative light and electron microscopy, and everything needs to be at the S3 biosafety level. Because if you want to explore corona, or I want not to use in vitro sample, I want really live samples from infected people, then I need S3 level. So we need a national center, and we need it yesterday. Because the entire world is buying these machines and is doing this. So if we don't get it yesterday, we are not going to be competitive. I know that I return to Israel and I can say hello to my lab and I love them very much, but I will tell them, sorry, don't have the tools to do the work. Maybe let's try to drive all of us to Germany every now and then to do the work. So, my 2 cents, and just want to finish wrapping up. What are the implications? So the implications are huge. We need this facility because we need antimicrobial drugs, not just for against the ribosome, new mechanism, antivirulence mechanism, and not just against the ribosome. We need materials for novel technologies. We need to understand communications between microbes, and some of them are by these fibrils, and maybe the most emerging thing, how microbes affect neurodegenerative and systemic diseases, and we need to understand all of that. And this is my beautiful lab in Israel, and I miss them very, very, very much. And they did all this amazing work, and thank you for inviting me to this beautiful conference.
Thank you very much, Meital. I hope that somebody will hear Meital and will do it. So. They are not here. So, Udi.
Yes, as I know, Meital probably it will happen.
Yeah, okay.
And I still want you to encourage you to stay in crystallography. There are beautiful things about crystal. Anat Bashan also told me that Ada is moving out of crystallography. But many of the organizations are related to the fact that it's like solid state biology, if I would say. But my specific question is about LL37. You know, I like this peptide because of the diphenylalanine motif, and you took it off. Why did you choose this segment?
Because it's the active core. It's the active core of LL37. It has the most amphipathic, so it has the highest vector for amphipathic nature, and it has 4 charges out of 6 in the entire protein, and it was shown in the literature already to be antimicrobial. So
is the full length?
It has a different spectrum. So LL37 is being cleaved in vivo to many active derivatives, and some of them are more immunomodulation, and some of them just work against different spectrum of bacteria. So this one is different. It's different from the full length, and it's also different from the structure that the full length can form. Itana? You choose.
Go ahead.
Hi, am I right? There's lots of evidence in neurodegenerative brains of oral decay, tooth decay bacteria.
Yes.
Is there some link to potential fibrils there, or the mechanisms of that believed to be different?
No, we think so, because one of the major bugs in periodontitis, forgot how it's named, but you know, infectious in your teeth. So one of the most abundant bacteria is secreting amyloid that is called the P1 amyloid. So there is definitely a connection.
Itana.
Can you actually knock out in the stuff? Can you knock out I don't remember the name of it. And
Yes. Yes.
According to you, it has to lose virulence.
Yes, it does. So people did this. It does. Yeah.
Yeah, one more down.
Vital, do you know about bacterial specificity of your antibacterial peptides? Are they gram-negative, positive, different
Yeah,
specific?
well, some of them are, we don't see a trend. Some of them are only against gram-positive. Some of them are against both. So we are trying to understand more of this by using different lipid mimetics, membrane mimetics with different compositions, so we can understand more about the specificity, because, you know, we are not a microbiology lab, so we have a favorite 4 bugs that we always use. So I don't really know.
No more questions. Thank
Thank
you
you.
very much, Meital.